So good morning, everyone, and thank you for joining early morning talk today. So we have Dr. Minna Holopainen from the lipidomic units of Helsinki University. So Dr. Minna received a doctoral degree in physiology and neurosciences from the University of Helsinki in 2020. And her research topic was related to immunomodulation of mesenchymal stromal cells, their extracellular vesicles, and their metabolipidomics. She conducted the research at the Finnish Red Cross Blood Services and also started working at the Helsinki University Lipidomics Unit in 2018. Currently, she is a postdoctoral researcher at the same unit and runs the lipidomics co-facility services and method development. For today's talk, she's going to talk about lipid mediators in acute inflammation and its resolution. What, when, and where. So, Minna, the floor is over to you.
Thank you so much. Nice to see you all, familiar faces and names. Let me just share my screen here. And I hope you can see my presentation now.
Yes.
Good. Let me just put your faces somewhere. Okay, great. Let's start. So thank you again very much, Dulmini, for inviting me to give this presentation to you here today. So the topic is lipid mediators, and our focus is mainly on the inflammatory side of lipid mediators. So I work at the Helsinki University Lipidomics Unit, and first I will just say a couple words on our unit and our platform. Let me just take a pointer here. So we, lipidomics unit, belong to this Finnish-wide Biocenter Finland national metabolomics platform, and also we are belonging into the University of Helsinki High Life metabolomics platform. And here you can see our head of units is Reijo Käkelä here, and the rest of our team here. And so we analyze lipids and, well, which types of lipids? Well, we can analyze lipids from cell culture samples, tissue samples, from rodents, people, horses, even insects we have analyzed, and also different wildlife samples such as birds, seals, and fish. And we analyze membrane and storage lipids with mass spectrometry. We also analyze fatty acids with gas chromatography and mass spectrometry. And our newest field is the analysis of lipid mediators and with mass spectrometry. And I have to say some words of thank you to Professor Jesmond Dalli, with whom I visited already some years ago, but this visit really helped us to develop this method that we have here today. So thank you for him and his team in London. But today we're not gonna talk about mass spec really, but more on the biology of these lipid mediators and why they are interesting, in my opinion. So first we will look at lipids and lipid mediators in general, then go through the pro-inflammatory ones, and then to the pro-resolving ones. So what is a lipid? Typically, when people think about lipids, we think about, well, fat tissue, adipose tissue, and storage lipids. We also have membrane lipids. All cells have membranes that encompass them, and we have organelles and such. And in the membranes we have lots of phospholipids, and phospholipids have all this like PET group and the 2 fatty acid fatty acids attached to it as acyl chains. And you can see, for example, in here, the cell membrane, and these fatty acids can be cleaved away from the phospholipids with phospholipases. For example, phospholipase A2 can cleave them away and then release them into the cytosol. And there they are converted enzymatically, for example, 15 lipoxygenase, 15 LOX into an oxidized intermediate, and then finally into lipid mediators that are, you can think of them as like lipid hormones, local hormone-like mediators. And the function of these mediators depends on their, of course, from which fatty acid they are originally coming from. So the precursors of these lipid mediators are polyunsaturated fatty acids, so fatty acids with more than 2 double bonds in them. And a very common precursor for these lipid mediators is an arachidonic acid, which is an N6 or omega-6 PUFA. And that is the precursor of leukotrienes, prostaglandins, and thromboxanes that are pro-inflammatory mediators, but it's also a precursor of a pro-resolving group of mediators, lipoxins. Also, other precursors for lipid mediators are these N3 or omega-3 PUFAs, EPA, eicosapentaenoic acid, and DHA, docosahexaenoic acid. And these you can get from marine fish, and originally also from microalgae synthesize these very highly unsaturated PUFAs. And EPA is a precursor of E-series resolvins. It's also a precursor of prostaglandins, leukotrienes, thromboxanes, and lipoxins, but these are not as like potent as those mediators from arachidonic acid. And DHA is a precursor also for D-series resolvins, varicins, and protectins. Here I'm just mentioning these 3 PUFAs as a precursors for lipid mediators, but also other PUFAs are function as lipid mediator, or can be lipid mediator precursors. So this is just an example, and maybe the most common ones that people talk about. I also want to say some words about nomenclature before we get into the interesting business, because if you look at the articles, you will run into these many names and might be confused. So lipid mediators are also called oxylipins. It just means all the fatty acids that have been oxidized. So they're not necessarily bioactive, but just when they have been oxidized, then they can be called oxylipins. But lipid mediator, the name itself means that the lipid or the molecule is a bioactive, and it's an enzymatically biosynthesized. So it already tells us something about the function of the molecule. They are also being called eicosanoids. All the 20 carbon, all the lipid mediators coming from 20 carbon fatty acids, and docosanoids are coming from 22 carbon fatty acids. But why do I want to talk about these lipid mediators really? Well, they are, like I mentioned, hormone-like autocrine and paracrine molecules. So when they're secreted, they affect the cell of origin or the nearby cell. They're very short-lived. That's why they are like very local hormones. They are also very potent, so they have to be like inactivated relatively quickly because they are very potent molecules. They bind to G protein coupled receptors to initiate signaling cascades. They also activate certain nuclear receptors such as PPAR gamma. And even though some of these lipid mediators are formed constitutively, but the production of many of these is triggered by inflammatory conditions, which of course highlights their importance in the inflammatory cascade. And well, which parts of body you could find these? Where are they produced? They are really produced throughout the body. Of course, with tissue-specific production, spleen with lots of like immune cell activity is very high on in lipid mediators. There's also liver, fat tissue, blood in human milk. There's also lipid mediators and tears, for example. So almost everywhere. And they have been found from cells, but also from extracellular vesicles. So perhaps vesicles are a means of transporting these potent molecules throughout the body. And like I mentioned, immune cells especially produce these mediators. And the biosynthesis often requires a triggering effect of an inflammatory stimulus or an infection for these mediators to be produced. So now we get to the inflammation part. So I'm sure all of you know that acute inflammation is a vital defense mechanism that we really need. And it has its five hallmarks. It has these fever, erythema, or redness, edema, swelling, pain, and loss of function. However, a prolonged inflammation is not good for us, but a return to the homeostasis is needed in order to prevent the chronic inflammation. And before it was thought maybe that inflammation would just passively dilute its way away, and then the homeostasis would just come naturally. But nowadays we know that the resolution phase of inflammation is an active dampening phase, and it's orchestrated by multiple molecules. And it also has its five hallmarks: removal of dead cells and debris, restoration of function, regeneration of tissue, remission from fever, and relief from pain. And like I mentioned, many molecules participate in this, and our special interest now, of course, are the lipid mediators that are called the specialized pro-resolving mediators, or SPMs for short. There are also other molecules such as annexin A1, chemarin-derived peptides, and acetylcholine that also participate in resolution of inflammation. Now, a simplistic scheme of inflammation. What happens? At the beginning, we have this injury, infection, and then we get the go signals. The pro-inflammatory lipid mediators are being produced, and that leads to increased edema, and also the recruitment of neutrophils to the inflammatory sites. And at the peak of the neutrophils, neutrophil levels, we have the acute inflammatory phase, which actually is required for the lipid mediator class switching, which then again begins the production of the specialized pro-resolving mediators, so the SPMs, which then again decrease the recruitment of neutrophils to the inflammatory sites, but increase the levels of monocytes and also macrophages at the inflammatory sites. And finally, we can achieve resolution of inflammation. And like I mentioned, there are also other molecules in play in all of this, but now just our focus is on the lipid mediator part. And we will now look at the GO signals, the pro-inflammatory mediators, in more detail. Typically, people have also called these eicosanoids. If you look at especially older publications, this word comes from Greek. Eikos means 20 in Greek. So it means all the lipid mediators derived from 20 carbon fatty acids. And traditionally, when people talk about eicosanoids, they talk about the arachidonic acid derived prostaglandins, leukotrienes, and thromboxanes. But like I mentioned, also, for example, EPA is a precursor of all these. So yes. And the classical eicosanoids that people typically talk about are the 2 series prostaglandins and 2 series thromboxanes and 4 series leukotrienes. And like I mentioned, not all eicosanoids actually are coming from arachidonic acid, but there are different series of these prostaglandins. Series 1 come from this dihomogamma-linolenic acid, and then EPA was the other precursor of these 3 series prostaglandins. And then we also have 4 and 5 series leukotrienes. And finding of prostaglandins was very major in the field of inflammation, and the people who first observed or elucidated prostaglandins, Zoomedicum and Bengt Samuelsson, received a Nobel Prize for this, and also John Vane for finding the effects of aspirin into the prostaglandin biosynthesis, so blocking of prostaglandin biosynthesis by aspirin. Yes, and when we look at the prostaglandin biosynthesis in a bit more detail, so again, the fatty acids are esterified into the cell membrane, where they are liberated into the cell lumen, and there they are oxidized further by this cyclooxygenase 1 or 2 enzyme into these final products. We have PGE2, D2, F2 alpha, and PGI2 prostacyclin that are the main prostaglandins from arachidonic acid. And then these mediators are secreted out of the cells, and they have multiple different receptors, T-protein coupled receptors that they bind to. Here, as an example, PGE2 binds to 4 different receptors that have different affinities and different distribution in different cell types. And like I mentioned, this cyclooxygenase 2, COX1 and 2, are very important in the biosynthesis pathway, and they can be inhibited by these NSAIDs, so non-steroidal anti-inflammatory drugs such as aspirin, ibuprofen. And COX1 is a constitutively expressed enzyme, isoform of the enzyme, and COX2 is mainly the one responsible for the inflammatory part. So its expression is highly increased by inflammation. Leukotrienes, prostaglandins are more produced throughout the body in different by different types of cells, but leukotrienes are mainly produced by immune cells, for example, by macrophages, neutrophils, and eosinophils. The main leukotriene is the LTB4, which is very powerful neutrophil chemoattractant. And there are also cysteine leukotrienes that are like leukotrienes with an amino acid residue attached to it. And these have various roles. For example, in lungs, they work as bronchoconstrictors, and they also contribute to the edema formation and as eosinophil chemoattractants. All in all, leukotrienes have importance in asthma and allergic and other inflammatory diseases. Here again, we have a similar graph of the one very simplistic that I showed you earlier. So here we have a barrier break and then the infection, and here you can see the prostaglandins started to be produced, and many of these pro-inflammatory mediators have overlapping effects with pro-inflammatory cytokines and chemokines, and the predominant pro-inflammatory prostaglandins are these PTE2 and I2. And LTB4 was the very powerful neutrophil chemoattractant. And here again, we have this lipid mediator class switching, which I will tell you a bit more in detail now. So during this class switching, these prostaglandins are actually required in order to start the resolution phase, because they can inhibit the production of this pro-inflammatory LTB4 through the inactivation of its biosynthetic enzyme 5-LOX, and also they induce the lipoxin, the pro-resolving lipoxin production through the activation of their biosynthetic enzyme 15-LOX. So we actually need the acute inflammatory phase to get the production of the pro-resolving mediator started. And this leads to one issue with our drugs, such as ibuprofen, for example, because then we actually block the prostaglandin biosynthesis, and then we don't get a proper lipid mediator class switching, which can compromise the SPM biosynthesis. And this then again can lead to a chronic low-grade inflammation. Yes, and now we go to the pro-resolving mediator part. Like here, I show you the PUFAs that they are derived from, and majority of these are N3, so omega-3 PUFAs. We have EPA and DHA, and also N3 DPA, which is not on my list, because otherwise it would, or on this diagram, otherwise it would be quite complicated. So, and the SPMs we're talking are the ones in green, so the ones coming, ones you can see in here. And when the prostaglandins and all those leukotrienes were found decades and decades ago, these SPMs are relatively new. They were only found a couple of decades ago, so still a novelty, but very well already established. They're very potent in pico-2 nanomolar range, and also they're metabolized very quickly, which renders their detection a bit difficult. Also probably one of the reasons why they were found so much later than the other pro-inflammatory ones, because they are difficult to detect. And this Charles Serhan was the one who first first found them. And these SPMs are one reason behind the health benefits of these N3 or omega-3 PUFAs. And there are studies showing an increase in SPM levels after ingesting these supplements, both in humans and also in vitro. And also it has been found that in certain diseases, there are lower levels of circulating SPMs, for example, in severe asthma and cystic fibrosis. Also, in vivo models, the administration of these SPMs can attenuate to disease symptoms, such as sepsis severity or periodontal disease. Again, I show you this biosynthetic pathway diagrams. I'm sure all of you are very fond of them, at least I am, but bear with me. So here, again, we have the precursor fatty acid that originally has been liberated from the cell membrane. And that is metabolized, that is then converted with this enzyme 15-LOX through different steps into, for example, here, a resolving D1. And DHA can be converted to other types of resolvins as well, resolving D2, 3, 4, 5, and 6. This is just an example of that there are many different mediators under one family, and still there are many SPMs found still today. Like I mentioned, this is a relatively new field, so new mediators are still found today. And one very interesting thing about aspirin actually is that it can also trigger the synthesis of these pro-resolving mediators. So as you remember, aspirin blocked the prostaglandin synthesis by blocking this COX-2 enzyme. But what it also does at the same time is that it acetylates this COX enzyme, and then this enzyme can actually catalyze the biosynthesis of an epimer of these resolvents. Here, normally with this pathway with 15-LOX, we would have an epimer with 17S configuration here on this one hydroxyl group. But here with this aspirin acetylated one, we have an R configuration here. And the epimers are also in that way quite interesting because they are also degraded more slowly, and they thus have potentiated pro-resolving effects. But this the formation of these 17R RBD1, it's not only happening through aspirin. It can also occur naturally via the cytochrome P450 enzymes. Yeah, but this is a very, very interesting additional work of aspirin. And like I mentioned, these lipid mediators, these SPMs bind to G protein coupled receptors. Not that many receptors for them have been found. Some agonists have been found. There's a couple more additionally to this list. But as you can see here, there are quite many mediators that can bind to the same receptors. So they have quite similar effects also because of that. And also agonists, they can also bind to some of the pro-inflammatory receptors as antagonists to prevent their function. However, still only a handful of receptors have been found, so the work on that is still ongoing. Well, what do these SPMs really do? Well, you remember this figure? We got all the way to the lipid mediator class switching last time, and here you can see the rest, and I will zoom in a little bit to here. So here we have the class switching here, and when the SPMs are being produced, they contribute to the first act on many innate immunity cells. For example, they recruit these non-foglistic, sorry, I can't pronounce it, but like non-inflammatory monocytes to the sites, and also they promote the efferocytosis of macrophages. And efferocytosis is a phagocytosis of apoptotic neutrophils. And actually, this after engulfing these apoptotic neutrophils, these macrophages become even more resolving macrophages that produce even more of these SPMs and help in the generation of these resolved exudates. And SPMs also have effects to adaptive immune cells, adaptive immunity cells, and finally help in wound healing and reaching the homeostasis. In more detail, what SPMs have been demonstrated to do, well, they counter-regulate the production of pro-inflammatory cytokines and chemokines, and also down-regulate the inflammatory NF-kappa beta gene expression. They promote tissue regeneration. I think this was quite has been investigated in a quite funny way. So they have these planaria worms that they cut the head off, and then adding the SPMs and seeing how quickly the head grows back. Also, SPMs have been demonstrated to alleviate pain, enhance bacteria killing and clearance, and thus also lower the antibiotic requirements for bacterial clearance in vivo models. And also in vivo models, SPMs have been found to suppress cancer growth. And this leads us to the question of resolution pharmacology. So as I'm sure you also agree with me, the failure to resolve can lead to multiple diseases. For example, rheumatoid arthritis, inflammatory bowel disease, cardiovascular diseases. Here you can see the normal inflammation. It peaks and then it goes back and resolves by its own. However, in red, you can see another type of inflammation where there is no lipid mediator class switching, and it just stays here on as a chronic inflammation. But if we would have perhaps SPM-based therapies, then we could get it back to normal. And this resolution pharmacology as a term, well, it refers to a possible change from using immunosuppressive treatments to pro-resolving treatments. And like we discussed, the NSAIDs, the ibuprofen, for example, they block the biosynthesis of prostaglandins, and thus they might actually delay the onset of resolution, even though, of course, they help us feel better in many ways, but it might also have some adverse effects there. But in order to reach this resolution pharmacology, we would need stable analogs and mimetics of SPMs, because like we talked, they degrade very quickly. So perhaps some antibodies or something would be a bit more easy to make as a drug. And also the identification of receptors and actions would be very much needed, so that also those receptors could be targeted as like therapeutically. There are some clinical trials ongoing also using SPMs or agents like it, for example, in human periodontal disease and dry eye syndrome. Actually, now I haven't checked this in some time, so I don't know if there are more. I was going to check this for you, but now I noticed I forgot to update this. But you can also check from the clinical trials website. There you can find all the clinical trials, so it's nice to sometimes go there and browse, see what they have. Well, one thing how we could also kind of promote or have this resolution pharmacology is that, well, if all these mediators are coming from these N3 omega-3 supplements, could we just supplement those beforehand? There have been some like dietary studies. Some show promise in certain inflammatory conditions, but some don't. Of course, in these dietary supplements, the question of dosing and what else the people eat is also something to be controlled. So not very easy studies to do, and also the supplementation of these fatty acids wouldn't work if there's a disease with an issue in the biosynthesis of the SPMs, for example, a defective enzyme or something related to that. I'm already in the take-home message slide. I want to highlight to you the biosynthetic routes of the lipid mediators, because they are originally or coming from the cell membrane. Of course, different types of cells have different sets of enzymes, and that of course dictates what types of mediators they can synthesize. However, if they don't have the precursors, if they don't have the precursors for the SPMs or lipid mediators, then of course they cannot then they of course cannot synthesize these mediators if they don't have the fatty acids in the beginning. I'm sorry, I was sick last week. Yeah. Well, this is a good place to stop. I just want to... So
It's
sorry.
okay, just take a minute. It's okay.
Yes, I wanted to say that the cell membrane has a very important role in the inflammatory status of the cell, because all the lipid mediators are originally coming from the cell membrane. Okay, and now I say thank you.
Thank you very much, Minna. And now the floor is open for questions. Until the listeners or the get it ready for the questions, I have one I have actually a few questions, but this is the first one I would like to ask. As you mentioned that SPM, like the IBD conditions, can be due to failures of the resolution, resolving, like you were talking about this IBD. As far as I know, it's IBD is one of the very difficult condition to detect. Like the early detection is quite difficult because it can be mostly misunderstood with other smoking side effect or maybe gastritis or something. So do you think this SPM threshold levels, like detection of certain SPM threshold levels in the circulation system could be used as a detection, like early detection level or method?
I would say that it's perhaps a bit challenging. I think it's I haven't like I would have to check, but I would think because the SPM levels can also go down in many different types of inflammatory conditions, then it wouldn't be that sensitive way of detecting only that IBD. So there, it's more like something we can then like see later when patients have been diagnosed with it. But unfortunately, I don't think it could be used as like a tool to screen the patients. Yeah.
Yes, Iresa, please go ahead.
Are lipids also like proteins? I mean, in the sense of proteins, we know they are species specific. For example, we have certain proteins that are common between different species, but they have individual differences. Like insulin can be from human, but with high homology, high similarity to what is also in cattle, for example, or in other species. Are lipids also like that? I mean, would a particular lipid that we get from animal or algae or plant or the others, would they be the same as the ones that we have in human, or would they differ a bit, or would be different?
I would say they are quite the same. Of course, different organisms have different types of lipids, but if like the same lipid, like for example, arachidonic acid is found from humans or cattle or fish, then it's like basically the same. But of course, there are so many different types of fatty acids, so there are different types of fatty acids. Well, mammalian like fatty acid profile is quite similar. Cattle have some like microbial origin fatty acids because they are ruminants. And but like in general, like the fatty acid profiling in that way is relatively like easy, because like we don't have like different isoforms of proteins or such, so there is not like that type of an issue.
And are there lipids that exist only in one species and not in the other species? And we can somehow use them to differentiate that where this particular material is coming from. Do you know of any lipid?
I don't know if there is like one species specific, but like I mentioned, like bacteria tend to have more like branched chained fatty acids, so we can like screen and see like, okay, well, these fatty acids are like from microbial origin when we see lots of branched chain fatty acids, because like mammals don't produce them themselves, so we can like have like certain types of like hints and such. And of course, looking at the fatty acid composition, we can get quite many clues on the biology of or the lipid metabolism of the cells. For example, if in cultured cells, it's quite actually easy and easy to see if the cells are grown, for example, deprived of these polyunsaturated fatty acids, because then they start to produce other types of PUFAs. They can't produce these arachidonic acid or EPA or DHA, but they produce these certain fatty acids that are not as good, but they have to produce something. So actually then we see an increase in these certain pathway of fatty acids. So we can get that type of information also from the fatty acids.
Yes, Suranga.
Yeah, thank you, Minna. So I just have a very quick question. When you compare with the EV fatty acid profile, EV membrane, and let's say from the origin, the cells which the EVs were origin, do you find a lot of differences, or are they the same? Like if you take like for example, like we use a lot of cell cultures. If you take the same, EVs enrich out of the same cells. Do you find a lot of differences, or are they very similar? Like if you compare the composition, especially, I know that people talk about the ratio of, especially the, I don't know, this PUFA ratio. So are they the same, or do you find big differences?
Well, typically I find in my previous studies, I found that the EVs tend to be perhaps a bit more enriched in PUFAs, because also, well, PUFAs, when you think about a cell membrane and then you have a fatty acid with lots of these double bonds, so it's a very bended, and it's like making the cell membrane also a lot more like labile. So those also tend to more like create the efflux. It's the efflux propensity of so like how much the phospholipid goes out of the membrane, and maybe perhaps those also like contribute to the budding. I don't know really what's the mechanism, but at least in our studies we found that there were more like they were more rich in the EVs, but in general the lipid profile of the EVs and the cells, they like are very like similar, but of course there are certain like EV specific differences, such as, well, there's a lot more sphingomyelin, or there's like more sphingomyelin there in EVs, and perhaps also certain like phosphatidylserine species and such. Yeah.
So there are differences. Okay, thank you.
Yeah.
Minna, do you think that the dietary or the lifestyle factors could influence the production of specialized pro-resolving lipid mediators?
Yes, I do. Actually, in some like there are some like older studies. I think the Inuits in Greenland and in Japan, where they ate like lots and lots of fish, that they actually didn't really have like cardiovascular diseases, really. But nowadays, when we have a more Western style diet, so with a lot more arachidonic acid in our diet, so I definitely think that also plays a role or perhaps one reason why we have more inflammatory based diseases nowadays. Of course, arachidonic acid is very vital and important for us, but now we're just getting too much of it from the diet, from like meat and
Maybe probably the last question then. Do you think that how specialized the pro-resolving lipid mediators being used as a targeted therapy, or then the drug development or intervention therapeutics in, I mean, like in upcoming years?
Yeah, I definitely think it's a hot topic, and yeah, definitely some efforts are already made towards that. And well, I don't know if we'll get like a similar breakthrough as like aspirin or ibuprofen. I don't know, but I know that maybe it's not a drug, but like I think you can already buy SPMs from the USA. So I've actually met this Charles Serhan, and he said that he's giving his kids these SPMs. So they're eating it already. At least he's a believer. Yeah.
Okay. Yeah.
So very interesting.
Thank you very much. Yeah. Is there any more questions to Minna? If not, I would like to conclude. Thank you very much for joining with us, and thank you, Minna, for your interesting talk. And we hope to see you in some other time also, and would like for the cooperations with your team. And thank you, everyone, for joining us with us today.
Thank you very much for
Thank you.
organizing, and
Yeah.
thank you very much.
Thank you.
Thank you. Bye.